Dr. Filipa Godoy. Filipa, could you please turn on your video? You are here, I believe. Okay. So, okay.
Yes, good evening.
Okay.
Hi,
Good
Safarina.
evening, Filipa. Hi, Filipa. Sorry, it's very late for you.
Yes, a little bit.
Thank you. And thank you for giving us the opportunity to listen to your presentation. Please, the time is yours. You have 15 minutes. Thank you.
See if I'm... am I sharing my full screen or my...
Yes,
yeah?
yes, it's
Perfect.
full screen now. Thank you.
Thank you so much, Safarina, for this invitation, and Gloria and the Microbiome team. I'm from the University of Puerto Rico. I'm a former student and a dear friend and collaborator of Gloria and her team. And I was requested to talk a little bit about the human microbiome and health. So I'm going to give a very brief overview in very different topics that I think are hallmarks of human microbiome research in the last decade. I have no conflicts of interest to declare. First, I just wanted to share a bit of what my lab is. We are at the School of Medicine, although we are a microbial ecology lab, and we run projects in the frontier of microbiology with ecology and genomics and metabolomics. And we look for cancer and immunotherapy, periodontitis, the microbiome of bees, sea urchins, soils, all in the Caribbean region, and that makes sense to the Caribbean communities. So when we talk about biodiversity or studies of biodiversity, we talk about microbes that belong to different groups of different domains. So the first knowledge came from the 70s, where Carl Woese defined the 3 domains of life. But then this idea has evolved through time. And so in 2016, the new tree of life is, as you see here in the middle, the group of Laura Hug and colleagues found that bacteria are really the biggest domain, with a candidate phylloradiation called CPR here, having, let me see if I can share my, anyway, I don't know how to share my mouse. You see my mouse? Well,
yes, here. I can see it clearly. Yeah. You are pointing to the middle panel.
Thank you so much. So now bacteria are like this big flower here, where this group is also a new group of bacteria called candidate phylloradiation. These are obligate symbionts, they're fermenters, and they, come up with very interesting functions that were before were not known. And then we have archaea and eukaryotes here in the bottom. And this last figure, with the emergence of the Lokis, of the Loki archaeota, which are this special group of archaea that are very, a monophyletic group with the eukaryotes. And you see here the slashing of the eukaryotes, like a very drastic change of the tree of life, and a new conversation that is, beginning to come up regarding 3 domains or are actually 2 domains. So this is still in discussion, but what I wanted to say with this is the basis of microbial diversity. And with this, I just wanted to share with you that, we live in a microbial world. There's all branches of the tree of life that are on our bodies, in us, on us. And this combination of microbes and humans, it's called the holobiont. This is a unit of evolution, which basically is shared through human evolution, through the distribution of, through the matrilineal lineage, through time, coevolution through birth in different human generations. Therefore, humans share a common phylogenetic signal. And we were not, this is not really recent. We have not been thinking about this for the last 10 years. About 100 years ago, already Mechnikov started thinking about what was the reason for Bulgarians to live as long as they did. And he associated that with fermented foods, with yogurt, and he found lactobacillus in yogurts. And he suggested that this was associated with their prolonged life and quality of life. He also was the first talk about bacterial translocation, inflammatory bowel disease, which did not have that name at the time. But with this, what we know is that the human microbiome has been developed since birth. So if you're born vaginally, you acquire a different microbiota dominated by lactobacillus that prepare your immune system. If you are born by a C-section, you acquire mainly skin bacteria, and this can disrupt the development of your human microbiome. And we also know that through modernization, lifestyle, different diets, stress, living indoors, drugs, these all can impact and perturb the microbiome homeostasis. And from this last, let's say 10, 15 years of human microbiome research, we know that every body niche is colonized by specific groups of bacteria, specific communities dominated by different taxa. And what I'm going to focus is some highlights on what we know about the gut, which is most of the research that has been doing in human microbiome is gut microbiota. And then if I have time, I'll talk about a little bit of why vaginal microbiomes are important. And of course, the benefits of gut bacteria are many. We can start saying that they have colonization resistance. This allows good bacteria to use the dietary materials and produce short-chain fatty acids and become this great barrier that does not allow other bad bacteria to colonize. They also synthesize vitamin K, which is great for coagulation, their barrier fortification, immune development, the metabolism of drugs and carcinogens, and even neurotransmitters, which helps in mental health. So there's this balance, this symbiosis. If we have communities that are diverse and they're contributing to this colonization resistance, or you have disruption and dysbiosis. And the first thing that I will say that was one of the first staples of what human microbiome research in the beginning, 2006, 2007, these first studies of 16S sequencing resulted is obesity. So obesity is a matter of microbes. Ruth Ley showed that obese mice have less Bacteroidetes compared to Firmicutes. And then about 6 years later, Dr. Jeff Gordon and Dr. Rob Knight made this beautiful experiment where they transplanted fecal matter from twins. One was obese, another was lean. And the recipient mice were gnotobiotic and acquired the phenotype of the donors. So microbiome recapitulated phenotype. Another thing that was very interesting is that the American gut, which is the biggest citizen science microbiome project in the world, has a lot of data, and from the data they have summarized the main findings. I would say that people that eat more than 30 types of plants per week have higher gut diversity and less antibiotic resistance genes compared to people that eat less than 10 types of different greens. And that's even better than if you're just a normal vegetarian or vegan. So what's important here is that you eat a diverse array of fibers, and they include kimchi, sauerkraut, and yogurt as sources of probiotics. And what they also showed is that bacteria, gut microbiomes that are dominated by bifidobacteria are more robust. And they also obviously found differences in industrialized, hunter-gatherers, and remote farmers in terms of their composition. Gut bacteria have also been, this is an immense effort through the years. As I said, I just tried to summarize what for me were the highlights and some things that I also teach here in my human microbiome course, is that gut bacteria also contribute to what we know of the gut-brain axis. First experiments treating mice with antibiotics showed that they have stress, anxiety, erratic behaviors. And then when they're given bifidobacteria or other probiotics, they're more calm, and bifidobacteria have been associated with anxiolytic effects. Also, people that have been under the taking many probiotics also have reported to have less sad mood. And even autistic children have dysbiosis associated with the production of propionic acid instead of butyrate. And changing their diets improves a bit of their life, their lifestyles and their health. Of course, I could not talk about human microbiome and health without talking about bacteriotherapy. Back in 2010, there was a case of a woman that was with Clostridium difficile associated diarrhea. She was in very bad shape, wheelchair, bloody stools, and it was only with a solution based on cow treatments of changing of rumen contents from healthy to diseased cows that this idea came up. And after a fecal matter transplant, as you see here, the patient modified the community very similarly to the donor at day 14. Then this became more complex. We know now that, for example, people with ulcerative colitis, they cannot just receive any types of donor stools. They have multiple cytokine assays to assess what types of stools are give a more inflammatory response to the host. So it's more complex nowadays, especially for these types of diseases. But then 2 months ago, FDA approved the first drug, which is a microbiota transplant called Rebiota. That, people take this drug after Clostridium difficile antibiotics in the first 24 to 72 hours. And that's pretty cool. So we have been advancing a lot. So it's an exploding field. Of course, we talk about probiotics, which are live bacteria, yogurts, kombucha, kimchi, kefir. And I mentioned to my students that these are the Kardashians of health, so they don't forget the KKK. And also, of course, the prebiotics, which is the most important thing, which is eat your fiber, at least 25 to 30 grams of fiber a day from veggies, from kiwis, and that have lots of polyphenols and very complex sugars, papayas, apples, and of course your broccolis and other veggies. Another very interesting finding that this was just last week is that you can treat methicillin resistant Staph aureus infections, nosocomial infections, through probiotics. This was proved with humans in a phase 2 trial in Thailand, where basically they show that 95% of Staph aureus disappeared from the stool and from the nose of the patients. And this is great. I mean, microbiome is really translational nowadays. So you can do not only characterize biodiversity, but really treat people in the hospitals in really serious conditions. I don't know if I have time, but just the last thing that I wanted to mention.
You still have 2 minutes around that.
Yeah, so remember I showed you the typical graph of less Lepson with the body niches, the gut being the more diverse and the vagina the least diverse. We don't really know if it's really the least diverse. Theoretically it is, but it can be very diverse in different regions of the world. Especially the proportions of lactobacillus are different for different ethnicities. So Caucasians have more dominated lactobacillus crispatus, which are more anti-inflammatory, that produce antibiotics. They produce oxygen peroxide. And then there's other types of lactobacillus that are not as protective. And for example, Blacks and Hispanics have more than of these less protective types. And the disruptions that can occur, like in the gut, the antibiotics and the diet and lifestyle, in the case of the vagina, are intravaginal insertion substances like douches, copper IUDs, soaps, and of course, infectious agents like human papillomavirus, which is the most important sexual transmitted infection, which occurs skin-to-skin contact. And there are more than 40 types in the mucosal areas causing cancer, cervical cancer, anogenital cancer, oropharyngeal cancer, penile cancer. So we've been looking at this and characterizing the diversity of HPVs in the Caribbean region. And I just wanted to summarize this by saying that women that have no HPV not even low risk, have a higher dominance of lactobacillus compared to those that do have even low risk HPV, going from a 25% relative abundance to as low as 2 or 3% dominance of lactobacillus. We also saw an increase in pH with age. So women in menopause can have a pH of nearly 6 compared to pregnant women, for example. Also, menopause women are at higher diversity, of course, with less lactobacillus. And we also see an increase in IL-1 beta and pro-inflammatory cytokine with more diverse communities with less protective lactobacillus. So basically, this human microbiome summary is that, ecological perturbations change irreversibly our microbiomes. We should eat fibers and probiotics, but most importantly, my message, especially for students, if you have student regional areas where you have groups of that want to study microbial ecology and want to learn bioinformatics, make groups and contact your associations and to work together with not only the Vault, but with other international groups that, give these trainings and like to have more collaborations happening worldwide. And I just wanted to mention Applied Microbiology International, which is based in the UK, but they also have regional offices. I'm ambassador for them here in the Caribbean. The ISME, the International Society for Microbial Ecology, which also has, each country is supposed to have one ISME ambassador. So if you're in Southeast Asia, or if go to ISME website and see if your country is represented. And if it's not, feel free to email me and I'll put you in contact with the ISME people. I know the ISME president is here, I believe. So he will be glad to have more regional efforts of ISME around the world. And I just add the example of AAAS because it's a representation of a chapter of the American American Association for the Advancement of Science here in the Caribbean also has a chapter, and we do conferences and workshops. And just to save the date, we have in Latin America, we have 2 important meetings this year. We have the ISME in the regional meeting of Latin America in Argentina in August, first week of August. The abstracts are still, we haven't even posted the deadlines for the abstracts, so we will be sure to share this with the Vault. And in March, we will have Dr. Blazer talking about the impact of antibiotics and the human microbiome here in Puerto Rico. So if you're here, please come by. So thank you so much.
Thank you, Filipa. Let's give applause to Filipa.